I'd like to welcome you all to the July meeting of the New England Comparative Effectiveness Public Advisory Council, where we will be examining Deflazacort, eteplirsen, and golodirsen for Duchenne Muscular Dystrophy. I'd like to begin by asking the CEPAC panel to introduce themselves.
Good morning, everybody. I'm Jean Ryer, New Hampshire Citizens Health Initiative at the Institute for Health Policy and Practice at the University of New Hampshire.
Good morning. I'm Austin Fracht. I'm the director of the Partnered Evidence-Based Policy Resource Center at the VA Boston Healthcare System. I'm also affiliated with the Boston University School of Public Health and the Harvard T.H. Chan School of Public Health.
Good morning. My name is Leslie Oakes. I'm with the University of New England College of Pharmacy in Portland, Maine.
I'm Steve Kogut. I'm a professor at the College of Pharmacy at the University of Rhode Island, where I lead our graduate program in health outcomes research.
I'm Teresa Fama. I'm a rheumatologist and the medical director of specialty care at the University of Vermont Health Network, Central Vermont Medical Center.
I'm Chris Jones, Director of Ventures for the University of Vermont Health Network and affiliated with the University of Maryland School of Pharmacy.
Good morning. I'm Rena Conti. I am an associate professor at the Questrom School of Business at Boston University, and I'm also the associate director of pharmaceutical economics and policy at the Institute for Health System Innovation.
Good morning. I'm Greg Lowe. Excuse my back. I'm a pharmacist and the program director of the Mass General Physicians Organization's Pharmacy and Econometrics program.
Good morning. I'm Martha Gold. I'm a senior scholar at the New York Academy of Medicine and professor emeritus at the CUNY Medical School.
Good morning. I'm Reverend Al Whitaker. I'm bivocational, so I pastor a local church here in Boston, and I also am a healthcare consultant.
Good morning. I'm Ed Westrick. I'm a primary care internist in Cranston, Rhode Island, and a former pharmacoeconomist.
Good morning. I'm Claudia Gruss, and I'm a gastroenterologist internist with the Western Connecticut Health Network.
Good morning. I'm Jason Schwartz. I'm an assistant professor of health policy and management at the Yale School of Public Health.
Good morning. I'm Lefteris Milonakis. I'm at Brown. I am head of infectious diseases there and assistant dean, and I also practice infectious disease and internal medicine.
Good morning. I'm Stacy Brown. I'm associate professor at the University of Connecticut in the School of Medicine in the Community Medicine and Healthcare Department.
Good morning. My name is Jason Waspy. I'm vice chair of CEPAC. I'm an assistant professor of medicine at Harvard Medical School, a cardiologist at Mass General. I direct our research program in health outcomes research in the Mass General Heart Center, and I'm a medical director of our physicians organization, Mass General.
And I'm Rob Azeltine, professor and chair of the Division of Behavioral Sciences and Community Health and director of Population Health Center at the University of Connecticut Health Center. I would also like to begin by having our panel of clinical and patient experts introduce themselves.
My name is Emma Cefaloni. I'm a professor of neurology and pediatrics at the University of Rochester in New York.
I'm Peter Kang. I'm Chief of Pediatric Neurology and Professor of Pediatrics at the University of Florida.
My name is Brian Denger. I'm the father of two boys who have Duchenne Muscular Dystrophy. Matthew, who passed away 6 years ago at age 20, and his brother Patrick, who will be 25 on Saturday.
Good morning. My name is Dawn Rose-Calla. I'm here today as a patient advocate, mother to a son diagnosed with Duchenne Muscular Dystrophy at age 4 in September 2015. We were also enrolled in Sarepta's 203 eteplirsen trials since he was diagnosed, and we've been receiving weekly treatment with eteplirsen, now Exon, since that diagnosis and enrollment. I also support, through advocacy and awareness, many different foundations in the Duchenne space, and am well versed in the Duchenne community, so I don't have anything specific conflict-wise to disclose or any financial conflicts.
My name is Mindy Leffler. I am the mother of a 16-year-old with Duchenne Muscular Dystrophy, and I also started a research organization called Casimir that's dedicated to capturing what we call patient and caregiver perception of change. We're in the process of developing novel outcome measures for Duchenne designed to capture and quantify ease of movement or difficulty of movement in patients with Duchenne as opposed to timed testing, which we think is more at the heart of the meaning of this disease. Those outcome measures are currently in FDA's qualification pathway for novel outcome measures and are in use in a number of clinical trials in patients with Duchenne.
Thank you. At the beginning of every CEPAC meeting, we want to ensure that all of our participants disclose any financial relationships with the health industry. All the members of the CEPAC panel do meet ICER's conflict of interest policy. However, we want to give anyone on the panel an opportunity to update their disclosures. We'd also like to give an opportunity for any members of our expert panel to disclose any relationships.
So relevant to the topics of today's meeting, I have to disclose that I have served on advisory boards as a consultant for both Sarepta and PTC Therapeutic, and I've also received support for clinical trials as a site investigator for both PTC and Sarepta.
The conflicts written down are correct for me.
So I serve on the Data Safety Monitoring Board for catabasis, as well as an external advisory committee for Reveragen.
Specifically, I actually serve as VP and a board member for Little Hercules Foundation, and I do other advocacy throughout, but not directly tied to any other foundation.
And as I mentioned, the outcome measures that we're currently developing are in use in several clinical trials. Companies that are developing drugs for Duchenne, including Sarepta, we're obviously pushing all sponsors developing drugs for Duchenne to use them because we think that they're much more sensitive to change.
Thank you. And all participants or public commenters during the course of the meeting today will be asked to disclose any financial relationships with the health industry. Before we begin the meeting, I'd also like to just call your attention to the ICER Code of Conduct, which is in the packets everyone received today. We're expecting a very exciting and productive meeting today. We're welcoming very diverse stakeholders, patients, clinical experts, advocates, policymakers, and we're really anticipating a very rich discussion, and we hope you'll keep that in mind throughout the course of the day. Thank you. I'd like to introduce Steve Pearson now, President of ICER.
Thank you, Rob. Sorry, good morning, everybody. Thank you for being here. It's a gorgeous day. Sorry we can't leave the blinds up the whole time. But thank you for being here. Thank you especially to those of you who've traveled a long way to be part of this in various ways. And thank you already. We'll get plenty of chance to thank you. But the participation from the folks on our, our expert and policy roundtable is going to be a critically important part of today's meeting. So why are we here today? Now, although the experience of every family, every boy with Duchenne muscular dystrophy is different, one quote that kind of captures a certain amount of why we're here today and expresses the unmet need of many families is here. The decline was robbing him of his independence. Instead of being an adolescent realizing independence, he was quickly relying on his family and mobility devices to help him perform any and every activity of daily life. His confidence and self-esteem were also quickly being compromised as a 10 to 12-year-old who was relying on others for all activities of daily life. He lost his ability to smile around the age of 12. What this captures in a small way is the experience of living with a terrible condition for which there has been no effective treatment. And many of us may have had family members or loved ones who've at some point in their life had a situation where there was a terrible kind of health problem and nothing to do, but precious few of us have had that happen to our children. And it really is a devastating situation for families. So we are here partly because we're at the dawn, perhaps, of an era in which innovation will bring new options for parents and their children. And that's very, very important. And we're here because there has been one recently, a couple years ago, approved drug at the FDA to treat Duchenne's muscular dystrophy, and there are others now in the pipeline. So one of the other reasons we're here today is because the day a drug is approved at the FDA, in the United States, the company gets to name the price. insurers get to look at the evidence, talk about it, and decide how they will cover it. And what rarely happens is that there's a discussion with the patient community as part of all that, for learning on all sides. And that behind closed doors, decisions are made about coverage and about pricing that are rarely transparent to patients, their families, and to the general public. So we're here today in part to try to open up that box and to hold a mirror up to the process and to use evidence as one starting point to understand how these decisions can be made for the best interests of patients today and in the future, with a realization that often price and access and coverage are linked in a way that can be very, very beneficial and can be very, very harmful, depending if we can get it right. So the historical context is that we don't always get this right. And obviously there's no one right answer. And so the ability to wrestle with this in public, we feel, is a very important process. Now, patients, we know, through this can have real difficulty accessing drugs, and not just in rare diseases. This is across the board, but it can be especially acute in an area in which the innovation pipeline is full of drugs going through accelerated approval, where there are lots of different kinds of surrogate outcomes and a lot more uncertainty, if you will, at the time that these drugs enter clinical practice. So the goals for today's meeting are to turn back to the evidence and to try to reflect on what we know from the published and other available evidence that we can bring to the table, and also be transparent and to try to talk about value. Now, value means different things to different people, but it definitely means trying to keep a broad view on the benefits, the disadvantages, the uncertainty, and the price, the cost over the long term, and how we pull that together in our thinking. So you've heard today the introduction of the members of the New England CEPAC. Let me just tell you briefly about ICER, since we're the kind of convening, if you will, body and the academic secretariat for this process. First, we are an independent, not-for-profit institute based here in Boston, well, over there in Boston. Our funding is shown here. We keep it updated on our website. This represents all of our revenue in 2019. As you can see, the biggest colored section of this pie chart is funding from a series of nonprofit foundations. The largest funder within that set is the Laura and John Arnold Foundation. We also receive funding from the Commonwealth Foundation, the California Healthcare Foundation, and several others. That's the money that we use for our research and this kind of convening activity. We run a separate program for policy summits and other kinds of evidence webinars that's supported by direct contributions from manufacturers in the life science community and by health plans and provider groups. And you can see those percents up there. But that money is used for that separate set of activities, and we have a very small residual government grant. So the report today that, in a sense, is the kickoff point for what we'll be talking about was developed over almost an 8-month period. And it starts with a full month of what we call scoping, and that's where we say to the world, we don't know yet what we don't know. And we have to talk to patients, patient experts, to clinicians, their societies, and certainly to the manufacturers and other stakeholders to get as firm a grip as we can on what the experience has been. What is the diversity of the experience of living as a family with Duchenne's muscular dystrophy? What is known and not known? What are some of the blind spots that we might run into when we start to look at the evidence? And then we do a deep dive, looking as kind of objectively as we can at the existing evidence, trying to encourage any evidence that's out there that we might not be aware of to be brought forward for public discussion. That process is twinned, if you will, by the development of a cost-effectiveness model, and in this case, it was done in conjunction with the University of Illinois at Chicago College of Pharmacy, and their faculty will be here to discuss the findings of the cost-effectiveness modeling. Throughout the 8 months, we go back and forth with the manufacturers, if they are participating in the process, and with public and patient groups to make sure that we are calibrating and getting comments and revision throughout. And then we have a very formal public comment phase on the draft version of our evidence report. Then we also have expert reviewers who are here with you today and had a chance to read the entire evidence report before it was finalized and put out. And then when we get to this meeting, it's important to talk a little bit about the conceptual background to how we're thinking about value, because it forms, in a sense, the structure to the report and also, to a certain extent, guides the discussion today. So we like to start our presentation, if you will, of our thinking about value by stating a clear goal that we really think stakeholders across the board in the health system share. And that is this idea that a fair price can drive and must drive fair access and provide adequate resources for future innovation. That this is not a zero-sum game, that we have to figure out how to keep all three of these moving forward. And for decision-making in the short term, we feel that there are two conceptual aspects that decision-makers need to keep in mind to try to support that goal. One, we term the long-term value for money, and the other is the short-term affordability, because sometimes those are in conflict. Not always, actually not that often, but there are cases in which treatments might have excellent long-term value, but there are so many patients in the short term that need treatment that the health system might buckle if we try to treat everybody all at once, that kind of problem. Here, we're going to be talking primarily about the long-term value for money, which itself we feel is obviously a composite idea. And to us, it is rooted in a firm grasp of our knowledge about the comparative clinical effectiveness. It starts there. And that doesn't just mean what's in the clinical trials. That's a starting point, but it is a root, if you will, position from which we start our thinking about long-term value for money. Building from that, we do create a model of the incremental cost effectiveness over the long term, because we want our health system not to be too narrowly focused on the here and now and the budget for next quarter or next year. We want a system that prioritizes and can sustain long-term value. Now, falling outside those two are other considerations. One, we label other benefits or disadvantages. And this is a very important, if you will, issue, because we know that clinical trials, especially those initially set up to gain regulatory approval, sometimes don't capture things that can be very important to patients and their families or the broader health system. So we'll be talking about those and having separate votes to make sure that we bring in those broader aspects of benefits or disadvantages into the discussion, because we want all decision makers to think of that as part of the package, if you will. And also important, in some ways you can have it as a separate bucket or you could have it encompassing the entire enterprise, there are contextual considerations. Every condition is different. Every step in the innovative pathway can be different. This is where we talk about social and ethical values and other aspects of the condition or the landscape that we feel should be important to take into some consideration in making judgments of long-term value for money. In our report, we focus in terms of the short-term affordability on a kind of an analysis of the potential short-term budget impact, but we let stakeholders make their own estimates of uptake and price for some in some situations, and we will be doing much less discussion of that in this particular context. So that's our conceptual value assessment framework, and I think it helps structure our reports, and in many ways it will structure the discussion and the voting that we do later today. The agenda is to move to a presentation of the evidence in just a second, and we have our clinical experts and patient experts here to be a resource to the CEPAC as they hear a summary of the evidence as it was presented in the report. We will have a formal series of public comments and discussion, followed by lunch, and then we will come back to do further deliberation on the evidence and to take the votes. So it's kind of a two-step dance here. We're going to talk about the evidence, wrestle with it, but we won't finish. We'll come back to it after further reflection and hearing public testimony, and then we'll do some further deliberation before we move into the voting, so that it's not just one step before we get there. Then we'll have a short break and have the culminating feature of our meeting, which is a policy roundtable. And during that, we will have this same set of experts joined today by 2 payer representatives. And that's where we try to say, okay, we've hashed out to the best we can what we think and know about the evidence and these other benefits and contextual issues. Now what? What do we do better going forward? What do we do better about coverage? Is there too much variation in how health insurance plans are covering something? How do we deal with that? What do we do about pricing? What influence is it having on future innovation, on the market today, on patients, on families, on the broader health system sustainability? And what future research and other policy recommendations can we make so that we create a better environment for fair pricing, fair access, and future innovation? How do we do that going forward? So this is a unique opportunity to have that black box again that usually makes these decisions opened up. And again, it means frank conversations. It means listening to each other, and it means trying to understand what we can do better going forward. So we're really looking forward to the meeting today. And again, thank you for being here. And with that, I will note that we will be done at 4 o'clock. We try rigorously to keep to that so that people can get out when they need to. You've already met our clinical experts, so I don't need to redo these slides. They've mentioned their interactions with industry and other folks, similarly with the patient experts. So with that, it's my pleasure to introduce you to David Rind, who is ICER's Chief Medical Officer, and he is going to lead us through a description of the evidence on the comparative clinical effectiveness. Thank you.
So I figure out the clicker. Thanks, Steve. I wanted to point out the team that worked on this, and in particular, I have Grace Lynn in bold there because she was the evidence author on this report and would be doing this presentation, except that she had a baby 3 weeks ago. And we, of course, knew that timing, so I knew I was going to be presenting today, but this would normally be her gig, and she's the evidence author on our report. We also have the ICER team, Felucio Agbula, Noemi Floich. Ifi Otoonye is no longer with ICER, but had worked on the report, and we have no conflicts of interest relevant to the report to disclose. So for background, and I know all of you have looked at the report already, but Duchenne muscular dystrophy is an X-linked neuromuscular disorder. It involves mutations in the dystrophin gene. Dystrophin is present in skeletal and cardiac muscle, and it strengthens and connects muscle fibers. And lack of dystrophin leads to progressive weakness, to wasting, and also to cardiomyopathy. Patients with Duchenne muscular dystrophy usually have dystrophin levels below 3% of normal, often much lower than that. There are other muscular dystrophies, including a disorder called Becker muscular dystrophy, and severe forms of Becker. It can have milder forms, but severe forms typically have dystrophin levels in the 5 to 20% of normal range, and that can lead to a less severe muscular dystrophy than Duchenne. Duchenne muscular dystrophy has a prevalence of about 6,000 people in the U.S. Symptoms are typically first noted at age 2 to 3. However, diagnosis is often not made that early and is often delayed until about age 5. And you'll see on some of the modeling that it used age 5 because of that. Early symptoms include falls, weakness, difficulty with stairs. Without treatment, loss of ambulation has typically happened by age 10 to 12, although there's substantial heterogeneity across patients, maybe because of different mutations, maybe for other reasons. Contractures develop, scoliosis, osteoporosis, fractures. Because of muscle problems in the GI tract, dysphagia, gastroparesis, children may lose the ability to eat. The respiratory muscles fail, and cardiac failure because it also involves cardiac muscle. Without treatment, without steroids, which we'll talk about some but clearly are of benefit, and ventilatory support, the death is typically in the teens or 20s. People live longer with better support, Supportive care includes physical and occupational therapy. Again, contractures are a big deal, maintaining strength. Skin care, the risk of decubitus ulcers. Pulmonary care, pulmonary toilet for people who are losing respiratory muscle function. Assistive devices, walkers, wheelchairs. Home and vehicle modifications, so that wheelchairs can get in and out, so that you can have ramps into vans. And then non-invasive and then ultimately invasive ventilatory support. Corticosteroids. They've been shown to improve strength, prolong ambulation, delay scoliosis and cardiomyopathy, and preserve respiratory function. The mechanism of benefit isn't actually all that certain. They're steroids, and there's thought to be an inflammatory component, so it's thought that that's probably how they're working, but people aren't sure. The appropriate duration of treatment is uncertain. Whether it's good to continue steroids after a complete loss of ambulation has occurred isn't so clear. And of course, steroids have the usual steroid side effects. The options for steroids basically include prednisone or closely related prednisolone or Deflazacort, Deflaza, which was approved by the FDA in 2017. And one of the main questions we're going to be asking today is the net benefit of Deflazacort compared to prednisone. We also have exon skipping therapies. Dystrophin is this very big protein. And in Becker muscular dystrophy that I mentioned earlier, you have mutations in the dystrophin gene that often lead to a shortened version of the protein that still has some function. And that gives the notion that if you could do something that allowed in Duchenne muscular dystrophy production of some dystrophin, even if it was shortened, that that could have benefit. And that's really the goal of the exon skipping therapies. Eteplirsen, Exondys 51, was approved by the FDA in 2016. And as you can tell by its name, it skips exon 51. And that can be used for mutations in exons nearby. Golodirsen skips exon 53, and an FDA decision is expected next month. And the other questions we're trying to answer today are the net benefit of using one of these exon skipping therapies in patients who have mutations amenable to that particular exon skipping therapy, in addition to standard care, including steroids. As Steve mentioned at the beginning of this and all through, we talked to all sorts of people and to lots of patient groups. This says patients. I have to say we talked more to parents than we did to patients. We heard concerns in different realms. In the realm of research, we heard concerns about the heterogeneity of the Duchenne population and that we don't have great natural history data across the various mutations and the variability that can occur, which gives people concerns about generalizability of results. We heard concerns about lack of validated measures for outcomes that reflect function, that reflect things that actually matter to patients. And you'll see some of that, but this was an ongoing concern that the measures currently being used, and we're hearing from our panel that we're looking for better measures, is a big deal. Perhaps it's obvious, but stabilization and slowing of decline are potentially really important outcomes. We don't have to necessarily take 5-year-olds who are slightly weak and make them perfectly healthy. If we could stop the disease in its tracks at age 5, that would be a huge win. And patient groups were very clear on that. We heard that before-after video data for the sorts of changes and differences that might matter in patients might be preferable to the sort of outcome measures that are currently being used, and that might be a better way of capturing things. We heard concerns about the durability of treatment effects. Of course, we don't really have long-term studies for the most part, and always concerns about side effects, particularly around the steroids. We heard financial concerns. We heard about the high costs of therapies and the difficulties getting coverage, and the uncovered costs for making modifications to homes and vehicles, that these can be a real burden to families. And again, this is a progressive disease, so the sort of modifications that you made 2 years ago may not be adequate today. We heard about caregiving burden anxiety, depression, isolation in caregivers, and that the burden increases as children lose the ability to ambulate. We heard that in terms of side effects of steroids, weight gain is a really big deal, that in people who are weak, being heavier can cause sooner loss of ambulation, that there's an increased risk of fractures, that it's increasingly difficult for caregivers if they're doing lifts or transfers and the patient weighs more, that can be a big deal. And then we heard that being Cushingoid or obese has psychological harms for patients, and so that's a big deal as well. I'm going to move now to the clinical evidence. So first we're going to focus on Deflazacort versus prednisone. And we have 3 randomized trials. By far the largest and best is the Griggs trial, which here says Griggs 2016. It had 196 boys, mostly had Duchenne. 4% of them may have had Becker. And that has to do with the fact that this trial actually was carried out in the 1990s. And there were less good definitions in the 1990s. And the results, nothing was really done with this study until the around 2014-15, when there was interest in getting approval in the US, and this trial was published. We have 2 much smaller trials. One is Karimzadeh, where only the patients were blinded, and Bonafati, which had just 18 boys with Duchenne. We also have 7 observational studies. I highlighted 2 of them. One is Shea from 2018, which was 114 patients in the placebo arm of a randomized trial. So it's a nested observational study, and that's nice because typically that does a very good job of capturing outcomes. And then we have McDonald 2018, and a lot of the focus that you'll see on the results and on the modeling is from that because it has a lot of patients for a long time. It has 10 years of follow-up, and that's not something that's that easy to find. In Griggs, the randomized trial, the primary outcome was muscle strength at 12 weeks. And I'm not going to explain what that measure is, but you can see that it went up by 0.27 in the prednisone patients. It went up by 0.15 in the Deflazacort patients, and despite being the primary outcome, no p-value was given for that in the publication. They did an exploratory analysis at 52 weeks, and you can see the numbers there, and they were not statistically significantly different. Bonafati, one of the other trials, found no difference at 1 year. Again, remember, that was a tiny trial, so it's hard to know what to make of that. In terms of motor function, and I put up there different ways motor function was assessed, Griggs didn't report 12-week results in a way that you could tell how Deflazacort did versus prednisone. Numerically, from weeks 12 to 52, results were better for Deflazacort on 3 of the outcomes, and the improvement in time to climb 4 stairs was reported as greater or with Deflazacort at week 52 with a p-value that is nominally significant. However, it's unadjusted, and again, this trial didn't get past its primary outcome as far as we could tell, so it's not clear whether this was statistically reasonable. But be that as it may, the results for Deflazacort look better at a year than they look compared to prednisone than they looked at 12 weeks. Karimzadeh found better function at 12 months as well with Deflazacort, and again, Bonafati, very small trial, no difference. Loss of ambulation is obviously a huge deal. We really only have this from the observational studies. McDonald, the big observational study, had a 3-year difference essentially in loss of ambulation. People continue to ambulate 3 years longer, statistically significant. One other observational study found a number not very far away from that, and then a 3rd observational study found minimal or no difference in time to loss of ambulation. In terms of harms, and this is a big deal again with the steroids, most evidence suggests that there's greater undesired weight gain with prednisone than with Deflazacort. In Griggs, it was 35% versus 28%. Most evidence suggests that there's a greater growth reduction with Deflazacort than with prednisone, that the boys don't grow as much while they're on it. And it may be helpful to talk with families about whether that's a good or a bad thing. You could imagine that in either way. Behavior change. These are, kids. You're giving them steroids, all the manic effects of steroids and irritability. Behavior change is a really big deal to parents. And there's a sense that Deflazacort is better on this. The evidence is quite thin, though. In Griggs, 14% behavior change noted with prednisone versus 9% with Deflazacort. Really hard to tell. And there's some suggestion of higher cataract rates with Deflazacort. So controversies and uncertainties. Well, we've got these observational studies, but again, we've got this one large study, which was a US-based study, although it was multinational. And in the US, at the time this was being done, Deflazacort mostly could be obtained by importing it and paying for it yourself. It wasn't being paid for by insurance. And so there's the concern of bias being introduced by the notion that families with greater resources were the ones who were able to get Deflazacort. And that's caused, we heard from all sorts of people, concerns about whether to believe that 3 years of additional walking as a result of that concern. In the randomized trial of Deflazacort, as I mentioned, we didn't have a p-value on the primary outcome. There was a sense of selective reporting in that trial, which seemed unusual. And then this business of behavior change. Again, this is a really big deal to know whether one of these steroids is better on behavior in young kids than the other. And there's lots of sense talking to parents that Deflazacort is better on this. I would just note that it's very hard for parents and kids themselves to tell things like this. If you think about the sugar experience, my daughter, who's young, insists every time she eats sugar that that's why she's bouncing off the walls. But meta-analyses of randomized trials suggest that that may not be true. And so this is an area where it would be really nice to have better evidence because it's very important to parents. All that put together, our sense was that the efficacy of Deflazacort seemed similar or perhaps better than prednisone, that undesired weight gain was probably less common, that growth retardation was probably more common. Putting that all together, we felt we had moderate certainty of a comparable or better net health benefit, and so a C+. At least comparable, maybe better. Then we'll move over to eteplirsen, which is the Exon 51 skipping therapy. And this is going to be this first study, the study 201, will be the only randomized data I'm able to present to you on exon skipping therapies. It involved 12 patients, 4 of whom got eteplirsen at its approved dose, 4 of whom got eteplirsen at 50 mg per kg, and 4 of whom got weekly placebo. There are no other randomized trials. There are 3 other studies in particular out there, 202, 204, and 301. 301 is the ongoing post-approval study, which has a comparison arm, but is an unblinded, and the comparison arm is not randomized. The primary outcome of 201 was dystrophin production. It was different depending on the dose. It was at week 12 for 50 mg per kg, and there was no significant difference compared to placebo. It was at 24 weeks for 30 mg per kg, and it went up by 23% in the patients who got that dose versus a loss of 4% in the people getting placebo, and that was different. The FDA had a lot of concerns about the way the assays were done in the 201 study, and they requested an additional study. This was done in 13 patients who were treated at the 30 mg per kg dose. 12 of them were considered evaluable. After 48 weeks and 30 milligrams per kilogram, on average, dystrophin increased from 0.16% of normal to 0.44% of normal. Out of the 12 patients, 1 patient got up to 1% of normal, and 1 patient got up to about 1.5% of normal. Nobody else got up to 1%. There were some functional results from 201. In the 50 mg per, this is the 6-minute walk test, one of those tests that we heard a lot of concerns was potentially overly gross in not noting small benefits. But in the 50 milligram per kilogram group, it declined by 0.3 meters. In the 30 milligram per kilogram group, it declined by 128 meters. And the placebo group, it declined by 26 meters, distance walked in 6 minutes. The investigators then said, well, 2 patients in the 30 mg per kg group declined really rapidly. We should eliminate them and only look at the 2 remaining patients. So having eliminated 50% of the patients at that dose, there was a 14-meter increase in walking distance in the 30 mg per kg group. That's the end of the randomized trial data. We have comparisons with historical controls in this and in other studies that found improvements in the 6-minute walk test and found improvements in pulmonary function in patients on eteplirsen compared with historical controls. For golodirsen, which again is going to be likely the FDA will make a decision on next month, we have a 25-patient study that really we just had an interim poster on that showed that baseline dystrophin levels were 0.095% of normal and increased to about 1% of normal after 48 weeks. We don't have any functional data, and we don't have any data on harms to relate to you. So limited evidence suggests minimal harms with eteplirsen, and I don't mean by that that it suggests there are minimal harms, that if there are any harms, they're minimal with eteplirsen, and we don't have data on golodirsen. In terms of controversies and uncertainties, the dystrophin increases with eteplirsen and golodirsen seem small. Again, there was this notion that if you could get up to those Becker levels, that would be helpful. These numbers aren't up that high, so if that thought going in was right, it would be surprising for this to work. Measures such as the 6-minute walk test can miss important outcomes. Broader measurements should have been performed. It would have been great if we had trials where people were measuring outcomes that were more relevant to patients. There are real discrepancies between caregiver reports and trial results. I would expect you'll hear some of that today from patients and patient representatives, where there's a sense that they're doing much better than what these results would suggest. Why is that happening? Well, you could imagine different reasons for that. One possibility, of course, is that this is a placebo effect, but other possibilities are we only have these really small studies. Maybe they haven't picked things up. And another possibility is perhaps there are rare patients who have big gains, and that while most patients don't benefit, we're missing big gains in small numbers of patients because we just haven't studied it well enough. So remember that those are all possibilities. The FDA had huge controversies about this drug. There were resignations from the panel. There were letters and fights that went up to the director of the FDA. And ultimately, the label for this, for Exon 51 says a clinical benefit of Exon 51 has not been established. For both these drugs, ICER felt that there were small increases in dystrophin levels, no moderate or high-quality evidence of functional benefits, and limited evidence on harms, but they appear safe. We rated the evidence as insufficient. In terms of potential other benefits and contextual considerations, if drugs are effective for DMD, we should expect reductions in caregiver burden, potentially dramatic reductions. The exon skipping therapies have a novel mechanism of therapy. These are really novel, different kinds of drugs. And then, obviously, this is a disease with a very high severity and lifetime burden of illness. We got public comments that we weren't using the right evidence standards, ICER's own right evidence standards for rare diseases. And part of that reflected a misunderstanding, I think, of our evidence standards. We don't have different evidence standards for rare diseases. We grade the same way. But we do say that we comment through the report in different places that it can be hard to generate higher quality evidence, and we hadn't done a good job of that in the draft, and we corrected that in the next version. There were concerns about the timing of the evidence review, basically saying you shouldn't expect that a drug that underwent accelerated approval would have better data. Exon 51 has been on the market since 2016. I would suggest to all of you that we could have better data by now. From our point of view, there's about to be another exon skipping therapy that may get approved. It would be great to know by now whether exon skipping therapy is or is not helping patients so that we could be making better decisions. And if it is helping patients, pushing for better coverage. And if it's not, moving on to the next therapy. There were comments that we needed to use real-world evidence more because there was a lot of heterogeneity. I actually think in situations where there's a lot of heterogeneity, having nothing to do with DMD in particular, that's where you really need randomized trials. When there's a lot of heterogeneity going on, randomized trials help balance that out for you. And so that actually gets at one of the reasons why randomized trials are so badly needed in understanding what's going on here. And then there was a comment that ICER is unable to review treatments for a disease like DMD. And you could take that two ways. One, that we're not capable of thinking about the evidence in situations like this. And I know many of the patient groups, and certainly this panel, were here where we looked at SMA a few months ago, and where Biogen had done beautiful randomized trials, and we certainly thought we were able to look at the evidence for that, and also look at the observational evidence on another drug for SMA and see what happened there. And people seemed to think then that we did a reasonable job. And then perhaps the other meaning of this is that we can't look at the cost effectiveness of this because in our draft report we had said that it was low value, and one of the people making this comment had listed a bunch of times that ICER had suggested that prices needed to come down to achieve adequate value, but didn't list therapies such as CAR-T for ultra-rare conditions, or emixizumab for an ultra-rare condition, or Zolgensma for SMA, where ICER thought that the treatments were cost-effective. So it felt like it was a selective comment that really any time ICER felt that a price was too high, it must be because we weren't able to do an adequate assessment, and we disagree with that. Thank you, and I'll take questions if there are some.
Thanks, David. I have a question about ICER's evidence rating of inconclusive, and on that slide, which is it's like slide 36, the first phrase to support that rating is that there are small increases in dystrophin levels, and just wanted to get your perspective on ICER's stance that there's a threshold by which increases in dystrophin levels translate into clinical benefit. That seemed to be a point of contention in some of the public comments. And, admittedly, the increases were very marginal, but yet might there still be a benefit to those increases, and perhaps they may influence the rating if it was interpreted as such.
Yeah, I think had we seen increases in the 5% range, even with the lack of functional evidence, we would have been more likely to rate this promising but inconclusive. And that those increases were so small pushed us, given the lack of functional data, to rate it as insufficient. Insufficient means insufficient. I mean, I think sometimes people are looking at that and saying, ICER said it doesn't work. We said we don't know. So, I wouldn't take the position that it's impossible that these dystrophin increases have patient-important benefits. We think we don't know, and we really wish we had better data. I'm sorry, I'm usually the chair picks people.
Thank you so much for the presentation. Thank you to Dr. Lin and the others for I know she's not here, but thank you for the work with the report. So one of the questions we're going to have to sort of deliberate about in this setting is that, at least referencing the comparison between the two steroids, is that there are 3 randomized controlled trials with severe methodological limitations and reporting limitations. And then there's a series of observational analyses. And so I guess I have 2 questions on that end. In the observational analyses, you referenced, and that resonated, that the thing we have to weigh is that in any kind of observational data, there's the possibility that other factors besides the different drugs affected the results in non-randomized observational data. And so one of those things was the Deflazacort could have been taken by wealthier families, for example, or better access to tertiary medical centers and so forth. Were there other, and this is going to be a critical point, I think, because of the limitations in the randomized control trials, were there other sources of unmeasured confounding or measured confounding that could have affected the results of those observations that were very clear, at least? I know it's always a possibility,
Yeah.
but was there something that you really popped out?
I'm actually going to turn to our experts here, in part because I think some of the buzz that we heard from experts was that they mostly didn't believe that there was 3 years longer ambulation with Deflazacort, but maybe you two have different feelings about that and feel that there's reason to think that those results are real.
Well, one other possible confounder is that both steroids, Deflazacort, but especially prednisone, they're used with very different regimens. So that is a little bit all over the map in terms of daily dosing, 10 days on, 10 days off, weekends doses. So when you compare the two drugs, you might also compare, different dosing. On the other hand, I think that a 3 years difference is a very large difference.
I think that's related to a comment I would make, which is I think it's been difficult to determine what the bioequivalence is at the doses that are currently being used, and that might make it difficult to compare them also.
Claudia.
Yeah, I guess I was about to ask a very similar question, and just to enlarge on that more, I would be interested in knowing if the biochemical differences and possible differing mechanism of metabolism between the two corticosteroids as well, and also what dosing regimen do you generally use for the two regimens, using the two different agents?
For prednisone, I tend to use somewhere between 0.5 and 0.75 milligrams per kilogram per day, starting daily, and then I explore one of the alternate regimens if the side effects tend to be more than tolerable. For Deflazacort, I usually prescribe 0.9 or close to 0.9 milligrams per kilogram per day.
Same here. So these are the recommended doses for the two drugs, and usually I start with daily dosing unless there are some exception, like if very young or side effects, and then you start looking at alternative high dose weekend and so forth. But generally the first step is the same.
Teresa.
Just continuing on this theme, in the randomized controlled trials, was there, I didn't see a look at for baseline levels of either drug at baseline, because there was some exclusion criteria. If you were on, prednisone for more than a year and things like that, you were excluded from the trials. But clearly, you could have been on some prednisone or Deflazacort during the trials, and I didn't see any comparison of what the baseline dosing, baseline characteristics of the dosing.
Are we talking about the Deflazacort trial or the eteplirsen?
In the Deflazacort trial. Well, either in any of the trials. In terms of just, when you look at the baseline characteristics of the patients in the trials, do we have any information on what the baseline dosing was of the corticosteroids?
So prior to the randomized trial, do we know what they were getting for steroids before they were randomized to the
Exactly.
I don't think we do. I'm looking over at Felucio, and she's saying, no, we don't.
Okay.
In the slide on the Deflazacort trial, in the C+ designation, it didn't include the increased risk for fracture with Deflazacort. Was that included in the analysis?
We included, so there are a number of other side effects that come up that weren't consistent trial to trial, study to study. There there is, and in your report, a discussion of fractures, and there was a retrospective study that showed a dramatically higher risk of fractures with Deflazacort, which isn't necessarily seen across every other study, but I think has to give people pause. Again, it would be great to have a long-term randomized trial so that we really knew the answer to that. I don't think we really know the answer at this point.
For the Etzler-Person study 201, the walking time results were 2 patients were removed and get the different result. Did the investigators go back and, for that smaller sample, look at the dystrophin production?
We actually had that question come up. As far as I know, they have not gone back and biopsied the children who did well or who did badly to see whether something different happened. Do anybody know otherwise? It's a pretty interesting question. I think it's an interesting question in general whether kids who are doing much better are having higher dystrophin levels. It looks like maybe you know something on this. Please.
So one of the things that I think is very important to understand in rare heterogeneous disease is the individual progression of each patient's disease trajectory at the time that they begin treatment. So, I mean, in my personal story, my son screened for that study, and he was considered too functional to be accepted into the study. And the reason for that is because the company at the time, had limited resources, and they only wanted to accept children that were on a decline trajectory, because then they thought that if they could reverse that decline trajectory, then the evidence would be more compelling. So as a result, I think they erred on the side of taking patients that were too precipitously declining, and there were 2 patients that, that were very, very close to the loss of ambulation entirely. And so I think kind of hypothesizing that was a responder phenomenon, there's a much simpler explanation, which is that those patients were beyond the point of recoverability for the outcome measure that was chosen to measure disease progression.
I certainly didn't mean to suggest in any way that the eteplirsen caused that precipitous decline, but that removing 2 out of 4 patients and doing an analysis of the dose that eventually got approved has its own problems.
No, absolutely, I don't disagree with that, but I think when looking at it on the face of it and looking at the simplest explanation, to me it is an example of improper outcome measure selection.
Sure.
And the necessity of choosing outcome measures that are very, very individualized to the patients that are participating in the study.
May I add to that? So just to add to that, there's an important study that wasn't mentioned here, which I mentioned in the introduction. My son was a candidate and a participant in. That's the Sarepta 203 study. Just to add to what Mindy said, by design, that study was a cohort for boys 4 to 7 years old. And the thought behind that was that they were developing study data for that declining period. I'll call it the age after 7, which would be more measurable. But the thought with this 203 study and why we chose to enroll was that it would give us data, which again hasn't been published yet, but give us data to point at exactly that, which is there's something to be said here about when this drug comes into play for the patient. And if we are diagnosing children between the ages of 3 to 5 on average, and you start our son on the drug at age 4, and our story is he started on that drug 3 months after diagnosis, then presumably there will be data that will come from this that will demonstrate that you will see changes in these children earlier, and it just changes the whole pathway for these children.
These are terrific comments. We're just a little behind schedule,
Sorry.
so it's okay, David. If there are no other questions for David, we should probably move on to the economic review.
Good morning, everyone. So I'm Surrey Walton. I'm from the University of Illinois Chicago College of Pharmacy and led the economic evaluation part of this report. I had assistance by Nicole Boyer from the University of Chicago, who's here, and one of my students, Danny Kwok. We received some money from ICER to support our time, but otherwise had no conflicts of interest. So the objectives here were to estimate the lifetime cost effectiveness of Deflazacort versus prednisone, and then of Deflazacort and supportive care, and then relative to prednisone and supportive care, And then also eteplirsen as an add-on treatment to corticosteroids and supportive care versus not having eteplirsen in patients that were eligible for eteplirsen. I'll say right off the bat that given the underlying evidence and what we had available to us, it was quantitatively most credible and we thought still relevant to policy to try to characterize a lower bound incremental cost effectiveness ratio for Deflazacort. So not saying exactly what it is, but searching for a lower bound. And with eteplirsen, given what was available and the cost of it, we searched for threshold treatments. So is there a treatment effect large enough that would make this cost effective? So that's what we did with our modeling. So I'll get into the methods. So the underlying data for DMD for an economic model kind of presents itself as survival curves. Kaplan-Meier results from clinical trials, and then kind of what is known about general mortality patterns across time that also act like a survival curve. So we built a multi-state partition survival model for that reason. We used US-based data because we wanted to be relevant to the US. For Deflazacort versus prednisone, it met the ultra-rare disease definition that ICER uses. So for that Deflazacort comparison, we use both a healthcare sector perspective and a modified societal perspective. And the modified societal perspective just means that we're not measuring all societal costs. We're being honest that we're trying to measure the available ones we have and being consistent with the second panel for cost effectiveness research recommendations to call this a modified societal perspective. But we're doing our best to be a societal perspective. So we use a lifetime time horizon. We use a discount rate of 3% per year on both costs and outcomes. The kind of cycle lengths or the way the partition survival model is used is based on kind of annual measures. And the primary outcomes are cost per quality adjusted life year, cost per life year, and we also do cost per year of ambulation gained. So this kind of illustrates the model almost exactly. So you can see that the health states that we have available to us are early ambulatory, late ambulatory, early non-ambulatory, late non-ambulatory, and dead. The most quantitatively driven part of this model is the time to loss of ambulation. That comes from these long-term clinical trial data and past efforts to synthesize those and fit survival curves that best characterize that. The mortality curve is based a little more loosely on historical data and kind of best efforts to fit a reasonable curve that matches basic patterns seen in mortality. And then the distinction between early and late in both those stages is based on historical data and guidelines that really existed before corticosteroids came into the market. But given the impact that's seen to best characterize the impact of corticosteroids and the loss of ambulation, kind of proportionally move those early and late stages to kind of match what they were in the past. So this was the best, we thought, way to characterize this given the underlying data. And the underlying data for costs and utilities are based on these definitions of early and late ambulatory, early and late non-ambulatory, and dead from again, past survey efforts in this field. So this shows you the doses that we used to do the costing. So for prednisone, it's 0.75 milligrams per kilogram per day. For Deflazacort, it's 0.9 milligrams per kilogram per day. And for eteplirsen, it's 30 milligrams per kilogram per week. And you can see these are weight-based. So in the model, the annual costs do vary a bit through time. We used average weights for kids by different ages, but the dosing caps off at 40 kilograms. So most of the costs are for a 40-kilogram kid. So again, we thought it was, again, the most credible thing to do, given the available data, but still of interest to policy, was to look for a bound on the ICER for Deflazacort, given its current cost. So the largest treatment effect that you can see in the data, which may have some selection bias in it, but is 3 years of gain to time and loss of ambulation. So we took that as a credible upper bound, and then we shifted all the curves by 3 years in the model to get an estimate again of this upper bound. There is no data on whether the proportionate amount of early and late changes with these drugs, and we felt, again, it was a credible upper bound to just shift all the curves by that amount. So that's what we did. There's a lot of different possible measures of serious adverse events. The study that we used for the treatment effect had measures of serious adverse events. So to be a little bit consistent, we just took their measures of serious adverse events between deflazacort and prednisone, and where there were significant differences, we tried to include that in the model. So this just kind of, it's a little hard to see. I wish I could blow this up more. But this just shows for prednisone and for deflazacort what we used to kind of build the model. And the deflazacort curves, again, are just shifted in parallel by 3 years. So we had patient utility scores and actually caregiver utility scores, which we don't use in the base case, but we use in some scenario analyses from an earlier study by these health states again. The patient utilities come from proxy measures using the Health Utility Index in the US, and the caregiver utilities come directly based on the EQ5D, and these are converted to utility scores. Here's the treatment costs. So if you take current available prices and based on the dose, and again, this is for a 40 kilogram patient, so they do vary by weight, but for the 40 kilogram patient, which is what you most see in our model, because they kind of reach it pretty quickly, prednisone costs $550 per year, deflazacort came out to $81,400 per year, and eteplirsen came out to a very substantial $1 million per year. We did try to validate those measures with payers and got pretty good confirmation that we weren't too far off. This just shows, again, from underlying survey data, the supportive care costs. So, in addition to the drugs, from a healthcare sector perspective and then from the modified societal perspective. And note that the modified societal perspective costs include the health sector costs. So the societal perspective is just broader, so it includes everything in the healthcare sector, and then plus some additional costs. And there were, the survey was pretty good about trying to be broad and incorporating the societal level costs. This just shows the annual adverse event rates. And this is just the serious adverse event rates that in the McDonald study, there were differences between deflazacort and prednisone. They had sort of 10-year prevalent rates, and we converted these to an annual rate that would lead to those rates after 10 years. So that's where these kind of come from. The costs largely are unavailable in the literature. Where there really was nothing, we put in a physician office visit cost. Where there was, some evidence for costs, we incorporated it. So for fractures and for cataract surgery, which is very rare but happens, that's why those costs are higher, because they were available. Okay, so based on all this, we get to the results. These just show the kind of overall totals in terms of cost and quality-adjusted life years, and years in ambulation. We shift these curves by 3 years. The difference doesn't exactly add up to 3 years because of discounting. You can see that in the societal perspective, the costs are much higher. The qualities are the same because that's how we did it. It comes out, so here's the incremental cost effectiveness ratio results. So for deflazacort, just kind of in the base case in the healthcare perspective, it's 361,000. And the modified societal perspective, it's a little higher, but very similar at 390,000. Again, we think this best represents a lower bound on the incremental cost effectiveness ratio. The societal perspective ends up being a little different just because the relative differences in costs between the early and late ambulatory and non-ambulatory are just a little different in the societal level than in the healthcare perspective. So they're higher, but the incremental changes are just different, and this is what, it could go either way, being higher or lower, and it turned out to be a little bit higher, but pretty similar. So we did some one-way sensitivity analyses. So you can think of this as the variance that would happen if one of the inputs in the model was changed, holding everything else constant. And this is called a tornado diagram, and the variables at the top are the ones where there's the most sensitivity to the input change, and those are the treatment shift, the early ambulation patient utility, and the Deflazacort drug cost. The rest of them, have a much lesser impact when they change on the model. There's no sort of credible range for these where the incremental cost effectiveness ratio gets even below 250,000. And again, this is already what we consider to be a lower bound. This is the healthcare perspective. We did this in the societal perspective, and it's almost identical. This is a probabilistic sensitivity analysis, varying all of the inputs in the model based on some reasonable guesses for possible distributions. And we found that in only 0.03% of the simulations was the incremental cost effectiveness ratio less than $150,000 per QALY. And again, this already was a lower bound. And when it got up to 500,000, most of the simulations were under $500,000 per QALY. So we did some scenario analyses to test some different assumptions into the model. I show a couple here. One is if you take a lower dose for Deflazacort and prednisone, the incremental cost per QALY gain drops to $283,000. And if you take the modified societal care perspective and you add consideration of changes in the caregiver utility for a single caregiver, that lowers the incremental cost per QALY gain to $202,000. And again, these are lower bound estimates. If you ask yourself, what would the annual cost have to be to make this lower bound estimate below a certain threshold? That's what this table illustrates. So given this 3-year treatment effect and so forth, for the base case results to be below $150,000 per QALY, the annual cost of Deflazacort would have to be $31,700, and some other values are up there for different thresholds. If you remember, the actual treatment cost was around $80,000. So this jumps to the threshold analysis for eteplirsen. So here we took the cost of eteplirsen as given, and we just projected using rightward shifts of those survival curves, potential treatment effects. That first line just takes those curves. Remember, for Deflazacort, we shifted it by 3 years. For eteplirsen, we said, well, what if we shift it by 10 years? What happens is, because eteplirsen has to be given every year, when you shift these curves, the treatment costs go way, way up. So you get more QALYs, but you get enormous changes in costs as well. So it leads to very large incremental cost per QALY gained. Even at the bottom, which is in the model taking a 40-year shift in these curves, and for patients that are on eteplirsen, giving them perfect health versus the utility scores that are there for the comparator, you still have it being over $1 million per QALY gained. So the limitations were that we had insufficient clinical trial data for the treatments modeled, and that's what pushed us to look for these kind of lower bounds or thresholds rather than an exact answer. Data were not available to build an additional granularity to the model, so we were limited to these early and late ambulatory and non-ambulatory health states. You could think of, with different measures, having more health states to look at with treatments. So the Deflazacort data were limited. And in addition, so we think this is a credible lower bound on the ICER, but even those inputs come from cohort studies, and there could be selection bias, as was discussed before, in those. We think that for Deflazacort versus prednisone, there's a lot of ways to improve our measurement and understanding of this. Any way you do it is going to almost certainly lead to a higher ICER than what we present. In the public comments, we heard that the model is too simplistic and does not capture the nuances of DMD. I think that's right to an extent. I think that the way we use the model and looking at a bound, you can have a more nuanced model and be more sensitive to potential treatment effects that may be important, but it's not going to lead to a lower ICER. We heard that the societal perspective should include caregiver utilities. For consistency with past efforts, we didn't do it, but we did do a scenario where we did include this. And the underlying utility estimates were not suitable for DMD patients. So I think there's room for important work to be done in measuring quality of life and utility more nuanced, in a more nuanced way for DMD patients. And the Health Utility Index may miss some potential treatment effects, but it is a validated and, widely used measure. The measures we got for utility came from the peer-reviewed literature. They have a lot of face validity. And again, we felt it was credible, certainly for the kind of lower bound thing that we were looking to do. So our conclusions were that the evidence for cost effectiveness remains sparse. However, given the best available information on this disease, compared to prednisone, and again, even as an upper bound, Deflazacort has a very high cost relative to the health effects that seem to be possible to be gained. And for eteplirsen, there really just isn't any plausible treatment effect that would make it look cost effective in this framework. So this should say questions, I think, but.
Any questions from the panel?
So I'm trying to do some mental gymnastics and figure out what the cost per quality would have been for Deflazacort at the cost that people were paying out of pocket when they were importing it compared to prednisone. I don't suppose you calculated that because that's all old news and over, but.
Well, I do not have an exact answer for that. My understanding is that those costs were a lot lower. It's a different perspective, right? Because that's a patient perspective being mixed with a healthcare sector perspective. But I'm sure from a societal perspective, again, the lower bound, it would look a lot lower. You could see in our model that the price of Deflazacort had a very large impact on the results. And my understanding that those potential costs of importing the drug were much lower than $81,000 per year. So possible that it would look cost effective.
So just to comment on the price, we had looked, I looked a couple of weeks ago at what it would cost to get it in Canada, and it was about $850 a year for that 40 kilogram patient. So if you could go up to Canada and get it, you could get it for $850, and it would be cost effective under these assumptions, clearly.
Again, the lower bound, which is what we're estimated, would certainly look cost effective. The exact cost effectiveness depends on exactly the effect you're getting. But it's a good point, and that's part of why we're doing the analysis, is to show, in the US, the way they're pricing it, what's the cost effectiveness.
Martha.
So you use a modified societal perspective, and I know there's a lot of disagreement in the literature about what to use here. But I wonder if you were to think about the productivity loss of caregivers, whether it would be useful to do a sensitivity analysis that looked at that. That might sort of shift things quite a bit.
Well, so that's actually in here.
It is?
Yeah, it is.
Loss of productivity. Okay,
Yeah.
all right.
Those indirect costs of the caregiver, at least as it was available in the survey data that we had in the US, which was a past study.
So indirect cost, so can you just explain to me what that exactly is?
They tried their best to estimate productivity losses to the caregivers.
Okay, thank you.
Ed.
I have a couple of questions about some findings that puzzled me a little bit, and I just want to make sure I understand this a little bit better. In slide number, 50, I was surprised to see that the caregiver utilities went up in late non-ambulatory stage. And I guess my question is for the parents. Is there something about, does life get better as the disease progresses? the caregivers?
So the boys reach a point as they get older where things are actually, they're stable. And many of them, especially in today's world where there's integration of technologies and social media, many of them reach a point where they're extraordinarily, they're happy. And if you do a comparison between them and their unaffected peers, I think you're going to find very little difference in their opinion of quality of life versus those peers.
Theirs is dropping throughout. It's the caregiver utility that goes up in the fourth stage.
You become used to it over time. I lift my son. He outweighs me by at least 40 pounds. It didn't come immediately. But I've learned a system of both working sleep deprived and maintaining a balance. And as a caregiver, I think that over time there's acceptance. You're seeing the individual doing well. Again, the quality of life from their vantage, and I think that those all add value to the family experience.
And my other question is on let's see, where is it? Slide 56. I was comparing the Deflazacort versus prednisone for the healthcare sector perspective and the modified societal perspective, and I thought that if they're spending 3 more years ambulatory, wouldn't those numbers be flipped somehow? Wouldn't you expect to see that number lower than the healthcare sector perspective?
So it actually can go either way quantitatively. It has to do with all the levels of the societal costs are higher right at each stage than in the healthcare sector perspective. And they increase across the stages, which is kind of expected. The absolute level of the incremental changes between those states are a bit different, and that's what drives the results. So we're pretty certain that they're right. I don't know what else to say. And so theoretically, it is possible for those to be different. It has to do with the incremental changes between the health states across the perspectives.
Can I just add one piece to that previous question? I think that when it comes, and I hate to kind of go back to the mantra of inadequate measures, but I think when it comes to assessing quality of life, and we've seen this in almost every rare disease that we've worked against or in it's that there is a desire to project a certain attitude that can confound the measurement of quality of life. And there's oftentimes a discrepancy between patients and caregivers in the way that it's reported. I mean, I will never forget a lunch and learn with patients with Barth syndrome, where the patient, was in his 30s and, oh, my life is fine and everything's great and I'm doing well and I can do this and I can do this. And his mother's jaw just dropped to the table and said, "You've had 5 heart attacks." So I think that there is absolutely this desire that varies significantly from family to family about the willingness to project a certain facade and to kind of reflect a certain attitude about the way that you live your life that isn't necessarily really reflecting that quality of life. And I will also say, just in terms of that utility score going up, that there is a large degree of difficulty during periods of loss. And when you have that fluctuation during periods of loss, it can be a much more difficult period of time emotionally than once the loss has occurred and you adjust to your new normal. So again, I don't think that's reflective of the difficulty in caring for patients at a certain stage of the disease, as much as it is the grief in the periods of loss and the uncertainty in those times of fluctuation.
Thanks very much. I have a question picking up or continuing the thread around caregivers that Martha raised, and it relates to the scenario analyses on slide 59. You show there what the incremental cost per quality gain would look like when you add caregiver qualities, did you do something similar to what the incremental cost per additional year in ambulation would look like when caregiver qualities are added to it to see how that would change?
No, because the caregivers don't get added years in ambulation, essentially. They have a utility gain that's associated with their kid being ambulatory versus non-ambulatory that's in there. But they don't themselves gain a year in ambulation, so it wouldn't change that.
But their utility doesn't look different in those stages in terms of caregiver utilities corresponding to different stages?
Well, I mean, I got to go back up to the utility scores. So the caregiver utilities are higher in the ambulatory states than they are in the non-ambulatory states. Not by a lot, but they are higher. So in that cost per quality adjusted life year, we add those gains to the gains from the patient. But when we're talking about years of ambulation, we're just adding up years that the patient is ambulatory. And so including the caregiver into that in terms of that measure wouldn't change it. Yeah.
Rena.
Thank you, I'm sorry. Sorry, I have a question about the pricing that you list on slide 51.
Okay.
So, Exondys is listed as having an annual price of $1 million, but the list price of the product is approximately $300,000, or at least at launch it was approximately $300,000. So what accounts for the discrepancy?
We have a price in terms of, per milligram, the price of the vials. And then we have the weight of the patient and we have the suggested dose. And that's what drives our calculation. I don't know what drives their calculation. We also have gone to payers and asked them, does our calculation look right with people who have had, use this right now? And they said it did.
I see. So perhaps it's an underestimate of the launch price that was listed at the weight.
It seems to be, and I don't know what drives that. I don't know whether that's a dose-based underestimate or a price per vial-based underestimate. Okay. Given the price per vial and the dose and the weight of the kid, that's where our calculation comes from.
Okay, that's great. Thank you. And then just another clarification, which is, so this is a product that is infused into patients in the clinical outpatient setting, correct?
Correct.
And so the discounts that are available for that product for purchase are not included in your price, correct? This is the reimbursement price.
Yeah. ICER has a method for applying a discount to the list price that goes with this kind of administration, and we used that. I believe it was AWP minus 15%. Varun can maybe comment directly.
Can you hear me? Yeah, so for all these hospital administered drugs, we use a hospital markup as well, which based on previous data, in this case we calculated as the wholesale price minus 15%, which is why you see the price that you see that we use. And like Sarah mentioned, it is for a 40 kilogram patient at 1 year.
Great,
so
thank you.
Cost.
Thank you. And then lastly, all of the administration costs are not included in this price, correct? They are included in the non-drug related costs?
We didn't include a separate administration cost in the model.
Okay.
So in that sense, it may underestimate the cost of the treatment a little bit. That's not something, given the magnitude of this, it won't change our conclusions.
Brian had a comment on the pricing.
Just a brief point of clarification. Exondys is available for home dosing for patients where it is appropriate.
Oh, that's fantastic. Can you tell me, is there a difference in out-of-pocket costs for the patient?
Yes, because you're not going into a hospital setting. And so many of the patients will opt for a Metaport, and through a nursing team, that cost will be different than going to a hospital infusion clinic.
That's great. Thank you.
I think we have time for, oh.
Sorry, there could also potentially be significant caregiver changes in cost, obviously going to a hospital, depending on the drive time and everything, versus doing it in-home dosing.
Thank you. Do you know what percentage of the population is opting to home dose?
I don't. We have time, I think, for one more question. Eleutherius?
I just want to mention my personal heartfelt, strong feelings about the families that are dealing with this. I think that I speak for myself, but I think that everybody can only try to imagine what you are going through and your boys, and just very strong emotions from here. Also, I was hoping that at some point we're going to have a more historical perspective of those medications and the pricing that was derived. Are those medications different from those that were available in other countries? Are they improved? The discovery happened here. It's just some perspective on understanding of how those prices were derived.
So these prices are U.S. prices. They are different internationally. I can't exactly characterize them. I know for Deflazacort, it's much less expensive in Canada and in other countries, presumably, than it is here in the U.S., far less. For Eteplirsen, I also don't know. Almost all drugs cost more in the United States than they do in other countries for a variety of reasons. But we didn't go and look at international costs.
Thank you. I think we should now move to public comments, and we have 4 individuals providing public comments. I'd like to call you to the main table. Fleur Chandler, Annie Kennedy, Mindy Leffler, and Christine McSherry.
If you need to use that.
Are we down a chair?
So we ask all public commenters to be mindful that we have 5 minutes each allotted to you, and we hope you can be concise and keep your comments within that range. We'll begin with Fleur Chandler, and if you could first state any conflicts of interest that you might have.